introduce our first speaker, who is Dr. Mwangala. I hope I say this right. Akamandisa, did I say it right?
That's very good.
So Dr. Akamandisa is one of the Global Genomics and Health Equity Postdoctoral Fellows, which is supported by Genentech. She's doing her postdoc work in Kate Nathanson's lab. And she got her, prior to Penn, she obtained a BA in biological chemistry at Wellesley College and a PhD in cancer biology at Emory University, where she studied the effects of PPMID mutations in pediatric brain tumor DIPG. So she's currently studying genetic susceptibility to breast cancer in women with high-risk disease characterized by early onset, family history, and or multiple primary tumors. And she is going to be pursuing these interests in the future. I don't want to say too much because I want to leave the time for you. So please go ahead and proceed, Mwangala.
Thank you for that introduction. And so just as mentioned, I am from Zambia, which is that country that's highlighted right there in Africa. And when most people think about African countries in general, they think of communicable diseases and just other infectious diseases. But there's actually a very growing incidence of cancer as well, hence my interest in studying and understanding cancer in African populations, and specifically breast cancer. And I'm particularly interested in breast cancer because it is the most prevalent cancer in women and the leading cause of cancer mortality globally. And so when you look at these results here, these are from the World Health Organization, we see that about 25% of all breast cancers happen about 25% of all cancers in women are in breast cancer, and the mortality as well is the highest of all the different cancers. And breast cancer has a lot of disparities that occur ethnically as well. And so women of African ancestry are more likely to be diagnosed with breast cancers that are known to have very poor prognosis, including at very early ages of onset. And so when you look at the number of people, this is data from the United States specifically, when you look at the number of people that have breast cancer at the age of 40, we see that the African Americans have the highest rates. That's the bar there that is represented in the brown color. And when we look also at a different subtype of breast cancer known as triple negative, which has the poorest responses to therapy and some of the highest mortality rates, the highest amounts also occur once again in women of African ancestry in the United States, is represented with the brown bar over there. And this is not only true in the United States, but even when we look more globally on the African continent, even though Africa, that is highlighted there with the black box, has some of the lowest rates of breast cancer overall, it has the highest rates of breast cancer related mortality. And this might be due to many reasons, including the fact that breast cancer typically tends to be diagnosed really late in African women on the African continent. But it is also known that African women have really, really high rates of that early onset breast cancer in the African continent as well. And it tends to be associated with mutations in BRCA1 specifically. And so even though there's not extensive data on the genetics of breast cancer in African women, it's very likely just based on the way that it presents that there are mutations that are contributing to some of this high mortality. There are also high rates of triple negative breast cancer. Some studies have been done in women of West African ancestry specifically, so in Nigeria and Ghana, and they have found that there's some pretty high rates of triple negative breast cancer, just as it is in the United States as well. And so just this, and so my interest in breast cancer ties in with genetics as well, because there is a very likely contribution of genetics to breast cancer in African women specifically. And so for that reason, I am studying genetics here at Penn. And so just bringing a brief introduction to the genetics of breast cancer specifically. In the United States, these are data from the United States specifically. Here in the United States, the average lifetime risk for developing breast cancer for a woman is 13%. However, there are some women that have an elevated risk for this. And these the risk can go all the way up to 80%, and it is due to mutations in different breast cancer susceptibility genes that could be high penetrance, such as BRCA1 and 2, or lower penetrance genes like CHEK2 and ATM. And it's not only the gene mutations that cause or that contribute to this increased risk, but then polygenic risk score, which is an aggregate of all the different risk SNPs that a particular individual has. Having a high polygenic risk score also tends to increase risk. And it can modify risk not only in non-areas of these high and moderate penetrance genes, but it can also increase risk in people that carry the high penetrance genes as well. And so if you look at BRCA1 and 2, for example, there is a pretty widespread that occurs for people with the low PRSs versus those with the high PRSs. And even in the non-carriers, people that have really, really high PRS scores have risks that are almost similar to people with moderate penetrance gene mutations. And there are different rates of mutations that are detected in different groups of populations of people with breast cancer. And so when you look in the general population, this is a very recent study, it was found that the rates of BRCA1 and 2 mutations in women with breast cancer in the general populations are pretty low, at just around 2%. And then other genes, they are around 3% and an overall 5%. And when you look at these other different high-risk populations, and so if you look at people with family history, and that is defined by having 2 or more relatives, first or second degree relatives, that have breast cancer as well. The rates of BRCA1 and 2 mutations are at 16%, and the rates in the other genes are mutations in the other genes are at 7%, with an overall of just around 23%. And people with early onset, and so that is people with breast cancer before age 40, the BRCA1 and 2 mutation rates range from around 10 to 20% in different studies, and the overall rates are around 19 to 29%. And people with multiple primary tumors, similarly, you have rates around 20 for BRCA1 and 2 combined, and then an overall of just below 30%. And knowing the mutation status of any particular individual is very important because it can inform different management for the carriers. If they have breast cancer already, then it can inform treatment options such as PARP inhibitors, which are effective in people that carry mutations in BRCA1 or 2 or CALB or CALB2. And then for people that do not yet have cancer or have only had cancer in one of their breasts, they could also take part in prophylactic surgery to prevent the incidence of new cancers. And for the carriers and their family members, there could also be enhanced surveillance and chemoprevention to prevent that breast cancer from
Can I
occurring.
interrupt you for a minute?
Yes.
I think when you change the slide, it would have looked like it's a bit cut off. I don't know why it is. So on the other side, so there were some people commenting that.
Oh, I see. They could not
The
see.
whole slide for some reason. Yeah, it looks like that on mine too.
On here, it looks fine.
Yeah, I guess.
Oh,
Yeah,
Okay, I guess it's for some people. Yes, in the comments, people ask.
okay.
Oh, I see.
Apologies, we're not quite sure why it looks fine here.
Yeah.
Some
Okay.
screens look like that, sorry.
Yeah.
Yeah, sorry. Right. And so even though we had these high-risk populations, we saw that the rates of mutation were not 100%. And so there are lower rates of mutations that are observed than what is expected. And so there is this missing heritability that occurs in high-risk breast cancer specifically. And so the question that I am looking to address and that our lab is looking to address is just generally, what are some of the genetic variation that could be contributing to this missing heritability, specifically in high-risk breast cancer, where there is a very high likelihood of inheritance of the breast cancer phenotype in families. And so some possible genetic variants that contribute to disease in the samples that test negative for the genes that are currently known in the current ways that they're measured could include, there could be mutations that are in genes that are not tested currently in different panels. And so these could be some novel susceptibility genes that exist that have just not yet been detected. There could also be possibly some mutations that occur in regions that are not typically tested. And so usually when people are tested clinically for mutations, it's mostly exonic sequencing that is done. And similarly in research studies, The main focus is again on exonic variants because they tend to be the ones that are most easily functional or functionally validated. And some regions that could potentially have this variation that are not currently looked at include some promoter and enhancer regions or variants in those specific regions. Or you could also have some deep intronic splicing that could occur that could again change the protein structure and function. And you could also potentially have some 5' and 3' untranslated region variants occurring. And then you could also have some structural variation, including copy number variants that are difficult to detect based on the current methods of sequencing that are performed. And you could also have some post-zygotic somatic mosaic mutations that occur that could lead to the development of cancer. In some diseases like tuberous sclerosis complex, it has been shown that some people that exhibit the phenotype do not have germline variants, but actually have some somatic mosaicism that actually leads to the tumor that leads to the tuberous sclerosis occurring. And so similarly, that could be a possible cause of variation. And then you could also have epigenetics impacting the phenotypes that are observed. And so we're interested in studying all of these different aspects. These are projects that are very early on very early on in the lab, and I'm just starting with some of them right now. And we'll be presenting just some of the very, very preliminary data that we've collected for a few of these different aspects, but we hope to look at all of them eventually. And so currently, the work that we're doing is looking at the first two only. So looking at novel susceptibility genes, as well as looking at non-coding variant analysis. And then I'm also going to mention briefly about some work that we're starting to do, looking at very early onset breast cancer as well, in which we think that the mosaicism might be playing a bigger role. So the approach that we have taken for identifying the novel susceptibility genes is twofold. The first one is we are a part of the Simplexa Consortium, which is the collaboration between Dr. Fergus Couch at the Mayo Clinic and Dr. Kenneth Offit at Memorial Sloan Kettering. And so the three centers, the Mayo Penn and Memorial Sloan Kettering, have collected samples from around the country. And it's a total number of around 11,000 germline sequencing of high-risk breast cancer patients. And the way that the people in the study were selected was each proband had to have either early onset breast cancer or multiple primary tumors, or at least 2 first to third degree relatives of breast, ovarian, colorectal, pancreatic, or pancreatic cancer under the age of 70. And there are some people that have that fall into multiple of these criteria within. And of these 11,000 samples, just over 600, so around 6,427 were sequenced here at Penn. And of these, 1,395 samples were contributed by Penn. So these are samples that were from patients that are here. And the sequencing that was done in this specific consortium is amplicon-based sequencing. It's a method known as QIAseq, and it was targeting 135 genes, including 536 SNPs that include some ancestry informative markers, so we can correctly identify people's different ancestries. And the second approach that we're taking for understanding the novel susceptibility genes is whole genome sequencing. We just got some of the results. We just got the results from the whole genome sequencing that we have performed. And so I don't have any data to show with regard to novel susceptibility genes, but it is something that we're going to be doing as well. And so one of the first things that we did when I first started was, as soon as the sequencing was complete, looked at the samples that were from Penn that we had other sequencing information on, so that we could see how well the sequencing was performing, because this was the first time that we had done amplicon-based sequencing, because prior we had been doing vector-based sequencing. And so of those samples that we had sequenced, 1,317 samples had passed our initial quality metrics. And of these, 300 had prior sequencing. So the first thing that we did was just to see are the variants that are being called in this new sequencing similar to the ones that we had seen previously in these samples. And we found that of the pathogenic mutations that were identified, we had a large number of them matching, so that was 64. But then we also had these new mutations that we're seeing in this QIAseq that we had not been seeing previously. And so in addition to having prior research sequencing, we also have clinical information for a lot of these patients because they are Penn medicine patients. And so we went to their patient charts to see who had, of people that we had identified mutations and who had mutations known in their patient charts. And we found that there were some mutations, a lot of mutations, actually 88 mutations in BRCA1 and 2, which are very high penetrance genes that we detected in our sequencing that were not recorded in the clinical database. And of these, 32 had prior commercial sequencing as well, which did not detect the mutations. And so this was very concerning because if this had been true, that would be a very, very big problem to have such high penetrance genes not listed. And so we thought there might have been something wrong or something happening in our sequencing because that was more likely than having actual clinical data be wrong. And so with that, we started looking, we looked at identity by descent, also just looked at distribution of different variants in our samples and ended up discovering that the entire cohort, there were some samples that had quality issues. And from this, this was about 2,000 of the 11,000 samples across the entire sample set that included 157 Penn samples that were not meeting the quality that was necessary. And so we were able to detect this problem that was a problem across the board and eliminated these 2,000 samples from future analyses. And for our samples specifically, we resequenced the ones that had been flagged. And when we resequenced them, we were able to resolve the mismatches that were occurring with the clinical sequencing. And so this was something good that we found out. And so currently, we are performing matching of these symplectal samples with controls from the Penn Medicine Biobank as well as the UK Biobank. We have access to both of these and have the data on site. And this is just showing, just very briefly, some of the PCAs that show the ancestries of the symplectal cases as well as the controls from the Penn Medicine Biobank. And so we're going to be able to perform good ancestry matched control cases. And after this, we're going to perform association studies to be able to see if we are detecting any mutations in or any associations in genes that we've not previously known. And the approach that we've taken for the non-coding variant analysis is looking specifically at intronic splicing analysis as well as promoter and enhancer regions. And the samples that we're using for this are the whole genome sequencing data that we just generated, as well as some analyses of previous data that had already been sequenced for different studies that had some whole gene coverage, not whole genome coverage, but was only looking at the sequence, the coding regions to understand or for the variants that were analyzed in the past. And so just a brief description of the samples that are, in this case, we had some early onset breast cancer, an early onset breast cancer cohort, as well as a family history cohort. And we looked at multiple known breast cancer susceptibility genes that are listed here. And the total number of samples after sequencing that we found did not have BRCA1 and 2 mutations were 515. And these are the samples that we're looking at for this analysis. So the first thing we did was looking at the splicing variant. And we used Splice AI, which is deep residual neural network that is able to predict whether every individual site is a splicing donor or acceptor, and whether a variant, when it occurs, whether it changes it. And the particular value of this specific method is that it's able to look up to 10 bases from the border, which allows you to actually look at deep introns, because previous methods were looking not more than 10 from the border, and so you're not really able to see deep within the introns. And so using this method, we were able to find a total of, we were able to find variants in known breast cancer susceptibility genes, as well as some genes that are not quite known to be associated with breast cancer. And so Interestingly, we found mutations in ATM and BRCA1 that are actually known to be pathogenic already. And this is data from ClinVar, which is the database that records clinical function of different mutations. And these had not previously been caught. And it was surprising that the ATM variant is just one base pair from the exon. And so it really felt like it should have been caught. But this data was sequenced in 2016, I believe, if not even earlier. And so it looks like just the technological advances that have occurred and calling up algorithms have allowed us to actually be able to detect new variants as well. The BRCA1 mutation is 5 base pairs from the exon, as it's a little bit more difficult to detect with the previous methods, but this is also a non-pathogenic variant, and so it was really good to see that we're able to find it. And the others don't really have known function yet, and so we are looking to functionally validate them. And so, as I mentioned, we're going to functionally validate using mini-genes the variants that are not known, or whose function is not currently known. And looking at that, prioritizing specifically the ones that have gains and are known breast cancer susceptibility genes. And that one family that I had pointed out that has the pathogenic BRCA1 mutation, that is 5 from the exon. When we look at the family, it does look like a breast cancer family because a lot of people do have breast cancer, as shown in that pedigree there. And the RAD51C splicing variant, whose function is not known, we looked at it as well and found that of the 3 variants that we found, all 3 of the people did have breast cancer, and they were all African American, and 2 are siblings. And so those are the 2 that are right there. And they are both affected with breast cancer. And so it looks like it's a likely contributing variant, but we're going to have to functionally validate it to be able to see whether or not it actually is associated with breast cancer. And then just our whole genome sequencing, we sequenced 24 samples that had very, very high risk breast cancer. I have the characteristics listed over there. And we compared the libraries using PCR-free methods and sequenced them on Illumina NovaSeq and obtained just around 30x coverage for all of them. We aligned them with the best practices in terms of BWA and GATK and annotated with variant effect predictor. We looked at splicing using Splice AI as with the non-coding variants, and then we also looked at structural variants using Manta annotated with ANNOTE SV. And so for the results, of the 24 samples, we did not find any likely functional structural variants, and we did not find any splicing variants either that had delta scores that were indicative of significant changes. But we did find a few variants that were in promoter regions as well as enhancer regions that we're going to need to functionally validate. But because we're seeing a lot of variants even within these regions, we had to come up with a strategy for how we were going to identify them. So we're restricting to known breast cancer genes for now, and then filter to population alternate allele frequency max of just under 0.1%. We're looking at variants that are heterozygous only, and the first thing that we have done now is looking at deep C analysis, which is another method that can offer scores that predicts the functional effects of non-coding variants. And 3 variants that have stood out so far occur 2 in BRCA1 and 1 in BARD1. And these all occur in either promoter regions or enhancer regions. And these are just tracks from USC browser showing that the variants are occurring indeed in those regions. And the score here just indicates what the deep C output thinks a particular variant does. If it's above 0, it says that in that specific class, there is an increase in activity. If it's below 0, there's a reduction in activity. So all of these reduced in activity, and the variants reduced the activity of promoter regions. And the variants themselves were found in either promoter regions or enhancers. And I just have 2 final slides here talking about that very early onset breast cancer. Some people have shown that people that have very early onset breast cancer, so that is talking about occurring in people that are 30 years old or younger. And in these people, there are much higher rates than seen in other populations of both BRCA1 and BRCA2 mutations. And then the only other gene that tends to be very highly mutated in these samples is P53. And so we looked in our population, and we found that we have 800 patients that have breast cancer that was diagnosed at age 30 or earlier. And of these, we have the sequencing information, either research sequencing or clinical sequencing of 300 patients. And this is the ratio breakdown. Unfortunately, people in the room cannot see it, but we have about 15%, about 16% African Americans with mutation that are of those 300 patients. And then looking at the mutational breakdown of this particular cohort, we have an even higher percentage that was in that previous paper, where just over 60% of people that are under age 25 have mutation, and just over 40%, so a total over just over 40% of those that are between ages 26 and 30 have mutations in all the genes. And as was seen in other publications, the rates of p53 mutations are significantly different in the people that are under 25 compared to those that are over 25 when we're looking at people that have breast cancer under age 30. And so we think that this is going to be a really good population to look at mosaic mutations, specifically in BRCA1 and 2 and p53, because they're just such high rates that occur. So in people that don't have mutations that are detected, we're going to be looking at mosaic mutations in here. And so some conclusions and future directions. And for our no susceptibility gene examination, we have so far cleaned our data for the Simplex in the Simplexa consortium and have prepared the cases and control matching, and are going to be performing the association studies. The whole genome sequencing is complete, but we've not yet done any analysis, but we're going to be doing analyses on that. And then for the non-coding variant analysis, using both the previously sequenced data as well as the whole genome sequencing, we have identified some previously unidentified splicing variants. I'm using that Splice AI. And we have also identified a few non-coding regulatory variants using our whole genome sequencing. And for both of these, we're going to need to functionally validate them. And then for the very, very early onset breast cancer, we have high rates of BRCA1 and BRCA2 mutations, as well as p53 mutations. We think that looking in these specific genes in this population for mosaic and structural variants is going to be something that will at least help us be able to detect them more easily. And with that, I would like to acknowledge the lab, as well as the Center for Global Genomics and Global Health for the funding these previous years. I would also like to acknowledge our other funding sources, and we'll be happy to take questions if Victoria holds executive.
Wonderful. Thank you so much. Well, can you try to start the video, and let's just see if that will work.
Okay.
I just want to see if the owl will actually turn on.
This camera looks like
Did
it's
it-
turned on.
Did it work? In the meantime, we would like to open this up to questions from the audience, including those of you online. Can you just go ahead and unmute if you want to ask a question?
Oh, now it turned on.
It's on.
The owl is on.
Yay.
Oh.
Okay. Any questions from people?
Is the camera in front of you?
Is it just me?
Oh, it's just you. Yeah.
Okay, that's okay. That's
all right.
okay. But if not, I have some questions about like future directions and some of the challenges, particularly of doing this research in Africa, because I know that's one of your interests.
Yeah.
So you mentioned at the beginning that I thought you said that the prevalence, that it wasn't known to be super high. Did you say something like that? But I thought that might just be because they're just not screening enough.
Yes, so it is both. And you're talking about prevalence of breast cancer specifically, right?
Mhm.
Yes, so it did have lower prevalence compared to the other countries, but it has been increasing, and it's predicted to increase even more. And so it seems that there are not just genetic factors, but likely epigenetic factors that are
It's
kind of
interesting.
causing the switch to happen as well. And so currently, I think initially, I would say right up until 4 or 5 years ago,
Mhm.
breast cancer was not the leading cancer in African countries, but now it is in women. And
Wow.
so it is increasing.
Right.
Yeah.
Yeah, I think that's going to be really important to look at. And so again, how many would you say, remind me, were novel, like that you found in the African ancestry data set that hadn't been reported?
So splicing variants specifically,
or any of them.
or any of them. So I think the splicing variant, especially the one in Red 51C specifically, was the one that has not been seen. I think
Okay,
all the samples that we found it in are African samples only.
Okay.
There was not any other variants that
right.
looked like that.
That's so, I mean, it shows how important it is to look at the diverse samples, but also to consider splicing, which a lot of people don't specifically look at splice variants. Okay, well, I think we have to move on.
Yes.
Thank you so much. Thanks again. Okay, Bege, you're up next.
Let's change that.
Yeah, can someone maybe try to help with getting, we're gonna try to, you guys online can see, but those of us here, we're having some
Yeah,
issues with the
exactly.
slides, so we'll just, and while you're switching that, I will go ahead and introduce Dr. Bege Dauda. So he is another Global Genomics and Health Equity postdoctoral fellow. And he is co-supervised by John Mertz and also myself. So he Bege obtained his PhD in biomedical ethics at KU, I'm not sure what that stands for, Leuven in Belgium.
Yeah.
His research is broadly focused on the ethical issues of research in developing countries. His interests are in the aspect of benefit sharing obligations in North-South collaborative research and the ethical and benefit imperatives of when you're doing global genomic research in Africa, with the aim of fostering the gains of the genomic revolution in the continent and reducing health disparities in global health genomics. He has, we're still setting this up, he has served as a committee member discussing data sharing issues in the African Academy of Sciences. And I think we're just about ready. There we go. Is it showing on the? It's so strange that we can't. Does anybody? Oh, swap displays. Go to swap displays.
But that would only swap it for them to see this one whilst.
No.
Oh.
No.
No, the swap.
Sorry.
Sorry, guys. Just give us a moment. We got to get this back up. So go back to the presenter mode. Presenter mode. Okay, and then up on the top, it should say swap when that comes on. Okay, swap displays.
Ah, okay.
That's how we've done it before. It seems to work. There we go.
Yeah.
And you guys can see it on
No, it's
there.
not shared yet.
It's
It's not shared. You gotta share it.
Yeah, so you need to do that first before you start sharing.
So share screen first. Okay, let's go. We'll see. Might have to be done again. Okay,
Okay.
awesome. Great.
Thank you.
Do you want to go ahead and turn the video on there?
Okay, sure.
Yes.
Okay. All right, awesome. Please proceed.
Right. Yeah, okay, so thank you for this opportunity to share some of my research work with you guys. And it's good to step down a little bit, from a lot of scientific work, to something more social, relatable in terms of ethics, right? Okay, so my research is basically examining the ethical terrains of GWAS in Africa. The main essence is, of course, we know GWAS is really important. We're talking about genomic revolution in the world. We need to keep up with ethics as well, not just doing research or genomic studies, but also the ethical part of research generally. Okay, so I'm going to have this outline to look at why is GWAS important in Africa, of course. And then, of course, looking at the study project outline as well as methodology that I used to have this, examination of GWAS study conducted in Africa. And then I'm going to give some preliminary results. It's not going to be like a whole lot of data out there because I'm still working on analysis of, the research or rather the data that I've obtained so far. And then I'm going to have some conclusion way forward. So these are points that, of course, a lot of you will be familiar with, know about. GWAS is important in Africa because of the fact that, looking at the uniqueness of African population in terms of knowing more of genetic associations, it's important to research or to have studies conducted in the African population. It is also very important to understand the complex nature of diseases, genomics and reducing health inequalities. And this is one of our quote, to support that by brought to me saying African genomes and the unique genetic structure of African populations harbor many clues to understanding human evolutionary history, which in turn can help shed light on disease etiology. So this is very important. It's important that we conduct GWAS in Africa. It is also very important to ensure that the continent is not excluded from the benefits of genomic revolution. Of course, we're in a period of genomic revolution. And if we, put aside or we push Africa to the side in terms of genomic study, we are trying to exclude them from gains of genetics generally, or genomics, advancement in genomics. So it is important to have that as well. But not just Africa, other parts that are lagging behind in terms of GWAS and other genomic studies as a whole. This is something that you'll be familiar with, certainly. I think this has resonated a lot among geneticists and researchers in global health studies in general, talking about the disparity in terms of GWAS conducted globally, global population. I always see this this picture similar to the concept of 10 by 90 gap, right, in global health, where you say just 10% of funding for research is spent on 90% of population in developing countries, and also the reverse. It's also true, that we spend 90% of health funding in just 10% of rich population, rich countries. Okay, so this is a huge problem, and of course, it underscores the fact that, or the notion that we need more GWAS and genomic studies generally in African population, but also Asian population. And so research is important, no doubt, but so is ethics. Ethics is also very important. So the call to increase diversity of sample in genomic research should help us to also activate the ethical compass of research in Africa. So in as much as we are trying to increase sample, to have diversity of samples globally, we should also be aware that ethics go hand in hand with research. It is important to juxtapose ethical conduct of research in developing countries in Africa. Why is it important? We want to avoid mistake of the past. And of course, there are a lot of asymmetric relationship, research relationship between global North and global South. You agree with me on that? Of course, we are not in the era, and I don't think that's going to happen, where Tuskegee can happen again. No, I don't believe so. Or Nazi experimentation. I don't think that's going to happen now in 21st century. But there are some new form of exploitation, that we do experience in research relationship. And in 2015, the European Commission coined a word called ethics dumping. This is a word that is coined to explain the new form of exploitation in terms of North and South research partnership. And there are different ways in which you can think about ethics dumping. The first one is, researchers or research sponsors in developed countries, they tend to circumvent, restrictive legal and ethical regimes, by taking research in developing countries, thereby avoiding very stringent legal and ethical regulations here, and they can also justify lower standard of ethical conduct in developing countries with the aim or with the mindset of saying we are helping people in developing countries. So lower ethical standards with the mindset that, the main aim is to help people in developing countries. That is a form of ethics dumping, or doing research, avoiding or not minding the structure or cultural structure in the country or in the local setting. So these are forms of ethics dumping, which, of course, it's important to address them. It's not just in genomic studies, but also in other biomedical research conduct. But I think it's also very important in genomic study. Why? Because in genomic studies, we don't really have an immediate, tangible product, from the conduct of research. One can argue and say, if you have a tangible benefit or a product generated from research, then you can accept some level of exploitation of people. That is debatable anyway, but one can put that argument, So, but for genomic studies or genomic research that we don't have a tangible product, then it's very important to, find a way of having this good research conduct with people, participants in those contexts. And it also ensures the incorporation of social values of research, among underrepresented populations. And by social value of research, think about research. The main aim of research is to generate knowledge, generalizable knowledge, right? But then we can't stop at that knowledge to say, well, we have, achieved a lot. We have understand some genetic associations, of a particular disease. There has to be an incorporation of the social value of research, which translates into what do people tend to benefit from the research, itself. You conduct research with, local people. What do they stand to benefit? What do they stand to gain? So it's important that, we put ethics in our research in order to ensure that. It is also important to ensure that ethics is not just trailing behind the genomic advances and revolution, right? This is something that ethics has been accused of. It's like ethics is always trailing behind scientific advancement. When scientific advancement is here, ethics is just trailing behind, to catch up. And then there's confusion everywhere. The Chinese guy that did that research that, it's an example, a lot of accusations that ethics was not really properly in place. That's why he was able to do that. So it's important to do that. Okay, so looking at the study outline. So with all that said, the importance of GWAS in Africa and the importance of ethics, we thought like it's important to conduct an ethical analysis of GWAS studies conducted in Africa. And the main aim is to just have this, viewpoint of the kind of transaction, the kind of research transaction, or the kind of partnership that we have, and then to look at it very well and address if there are any ethical issues surrounding the kind of research collaboration that exists in doing GWAS. We all know there's always this flow of research collaboration from Western countries to developing countries, and hardly the other way around, So what kind of partnership, does that collaboration, how does it go? It's important to have that. And in doing that, we're thinking of two ways of doing it. First of all, to have a feel of what the literature is saying about this GWAS studies conducted in developing countries in Africa. So we're going to do a systematic review, but also we feel there is an important part of qualitative interview study, which is a way of complementing the systematic review. This is something that is ongoing right now, the systematic literature review, and hopefully manuscript should be ready for submission in July. For the other part, this is potential. What I'm thinking about, like the study will be greatly informed by the systematic review, and of course, potential participants for the interview will be derived from the systematic review. So this is the general outlook of what I intend to do, the whole picture of it. So going specifically to the systematic literature review. So these are the things that I consider for my review. First of all, the study inclusion criteria would include just original papers containing primary or existing secondary data, studies that investigate measures of genetic association with single or multiple disease traits, studies conducted in country or collaboratively with other countries within Africa. So, it has to be studies exclusively done in Africa for it to be included in the study. Exclusion, we're going to exclude any multi-regional studies that include populations from other regions of the world. So it could be studies conducted with migrant population that are living outside Africa will also be excluded, and of course review articles. So what we did with the data search, so first we thought like it would be just, like very easy to just, go into GWAS catalog and have our search there. So we did that. What we did was just to throw all the names of African countries to see what will come up, in terms of papers and publications on GWAS studies. And unfortunately, examining the hits that we had, it showed that it's not quite exhaustive in terms of the papers that we're getting. And there were a lot of papers that combine research with other populations of global populations, not just in Africa, which of course is one of our exclusion criteria. So we had to look closely to select the papers that actually match selection criteria. So we did that, but then, we felt like it's not quite exhaustive in terms of the papers that were returned. So decided to check on PubMed. This is the search criteria that we developed to build. First of all, genome-wide association studies, or GWAS, these terms, African with African, and then we entered, the different countries of Africa. And we excluded animals and plants, genome studies related to animals and plants. And finally, we had this 436 hits. And out of that, it includes review papers, and so we had to go through everything again to select the ones that fit in our criteria. And in doing that, we came up with okay, the next slide. Okay, so we obtained the articles, all of them, about 64 of them. It's going to be on the next slide. But these are the ways that we processed the articles. We have everything transferred on spreadsheet. PDF copies were downloaded and saved in Mendeley. And then these are the excerpts, we coded this excerpt to capture this following nodes. Author's country, population descriptors, population choice justification, types of data, primary, secondary, IRB approval, study benefits, and the funding source. Now I'm going to talk about each of these points just to give you an idea of some of the findings so far. We're still coding, but there are some, inferences, well, not generally inferences yet, but some emerging results that are emanating from what we have done so far. This is supposed to come before that one. This is the total number of articles that were included in the study, 64. 31 from GWAS catalog and 33 from PubMed. Publication year for articles ranges from 2009 to 2021. Author's country, we also coded author's country of affiliation, which shows great diversity among collaborative research partners. So the idea is to have first author and the last author, just to see, the level of collaboration. But we thought this is not going to capture, the degree in which people are collaborating or researchers are collaborating for GWAS. So we decided to pick the names of countries in between the first and the last author, just to have this broad view of who and who is collaborating, So what we've seen so far is there is more intercontinental collaboration between Western and African countries, which is kind of expected because we know most of this funding comes from Western countries, and African countries are more or less the host country for research. But of course, we expect there should be some form of partnership with researchers that are in Africa. So yes, there is that collaboration between intercontinental collaboration. But one thing again that we've realized is we've seen less intracontinental research collaborations. By that, H3 Africa, for example, one of the major objectives is to increase the way researchers within Africa are communicating with each other and conducting research within the continent. So this is not quite encouraging, looking at that or with that, that mandate in mind, because it shows that people are not really, collaborating within the continent, and it also shows still the reliance of funding from Western countries, which I think as an ethicist, if we are to move towards research independence in Africa, then one of the ways that we can encourage that is to find a way of generating funding within the continent. That is one way in which we can move towards research independence in the continent. So there is less intracontinental research collaboration. So that's typically articles with no Western collaborators. In terms of population descriptors used in the articles, it varies. We've seen population descriptors like, racial categorization. For example, Southern African, Black, white, or colored. We've seen that a lot. Some are using typical country descriptors, Ugandan, Tanzanian, ethnic, Hausa, Yoruba, in Nigeria. One thing that caught my attention, I didn't see much of the use of the word ancestry, in the articles that I have seen so far to describe participants. I think it's, a little bit problematic within that big framework of moving from the use of racial categories to ancestry. And that's a bit problematic. But then, of course, we are not making any, drawing any conclusions for now as far as findings. Population choice justification. Most of the deduced justification for choosing the said population is associated with increasing diversity of sample. So you often see this quote. It says, for example, this one, the studies introduced above has mainly been conducted in cohorts comprising samples of European ancestry. And then there is need for us to conduct the same research in African population. Okay. All right. So this is what we've seen so far. I want to talk particularly on study benefits. So we are trying to look at all the articles to see, is there any mention of, any information that captures the idea of what this study is set to achieve? What is the main goal of the study? We try to capture that. And
Bege, just to let you know, we have to end pretty soon. So I don't know how many more slides you want.
I'm second to the last.
Okay.
Yes. Okay. So the idea of benefit is what the research is set to achieve, right? But then, of course, there is more like a whole spectrum when it comes to benefits, and on the one hand, it might be benefits related to scientific knowledge generation, right? So that's typical of, when you think of yourself, when you think about yourself as a scientist, you want to understand different genetic association, you want to increase your knowledge, you want to have, more novel associations and so on and so forth. So that is a benefit. But when you think of that benefit to the larger society, especially the people that you're conducting research, it does not really have any impact on them, So, and that's why, on the other hand of the spectrum, we can think of tangible products of the study, coming from the study, and that one, like I said, when you think of genomic research, we're a little bit far away from it for now because the nature of what we're doing right now in genomic study, we're a little bit far away from that. So between that spectrum, we have to think about ways in which people that we are conducting research with can be part of the research, just to feel that this research is for our own good, it's for our own benefit, because you don't have a product to give them to say, this is the end result of this study. But then when you negotiate, when you talk to them, when you incorporate them, community engagement and all that, then you make them feel that they are part of what's going on. So what we are extracting in the document is more or less to see at what level is benefits listed in the publication of the articles. So these are some of the findings so far, but there is a lot of articles that we need to code in order to understand more and for us to come up with discussion and conclusion. Okay, so conclusion, very imperative to keep watch of the ethical and societal value of research conduct as genomic research continues to proliferate in Africa. We need to keep track of that. I believe this study is going to contribute and suggest ways to improve the dynamics of genomic research collaborations and ensure that research is beneficial to collaborating partners. Very important. I think going back to the research outline, the other part of research interview would help us to understand even more some of the things, some of the ideas, or give us ideas of what is not incorporated in the articles. For example, people don't write about community engagement. How do they engage the community in research? So that's a good way to understand that. It's through interviews of authors that are involved in research. Thank you.
Thank you very much. We're a little bit over time, but if there's anybody, including those online, who have a question, can you check if there's anybody in the chat, or just jump on if you have a question?
There was a question in the chat for Mwangala earlier.
Okay.
But she had,
We'll have to, we'll get to that later.
oh,
Dan, you
I have
have,
a question about the
Yes.
GWAS survey. So based on your exclusion criteria, you don't consider papers that say have both sub-Saharan African and also African American or other ethnic.
No.
Okay.
Yeah. So the idea is not really about the population itself, but rather the conduct of the research of GWAS in Africa, within Africa.
I see. But I wonder, nonetheless, I mean, if a paper has, say, they collect novel or new data, right, from some African country, and then they use I don't know, for example, commonly in GWAS will do replication, right? And they may do the discovery GWAS and sub-Saharan African population, then they try to do replication in the African American population. I still feel like that's relevant for your research, right?
Yeah. What I'm thinking is it's important to know the dynamics about for those that are involved in the research itself in Africa, So,
but I think you're just saying that rather than excluding them from that list, maybe you can also include them. Is
Right,
that,
yeah.
that,
Because they're still doing, there's still that dynamic of they're collaborating.
right?
If I can weigh in?
Yes,
This is
please.
John, I mean, we're also looking, speaking pragmatically, we're also looking to limit the sample size.
yeah.
Yeah. Meaning that, what do you mean by that?
Oh, we have 60 some odd trials, studies as it is, and one person, I'm not exactly sure how many we would wind up having, but I think it was not an order of magnitude larger, but it was several times more studies that might have included some people from Africa, but also people from many other areas of the world. So it just became easy to kind of limit this assessment, especially because it's the first of its kind.
Right.
And because if they're only doing study in Africa, then they're going to be describing the methods that they used in Africa, as opposed to combining kind of methods across multiple continents and trying to figure out what they did where. It just turns into a real morass.
That makes sense. So I just have couple of comments regarding benefit sharing.
Yeah, we can talk later.
Regarding the benefit sharing, I mean, there are a couple of thoughts. Some people are talking about, for example, in the case of commercialization, because if you really want therapeutics to be developed, then it's going to be pharmaceutical companies who are going to do it,
Mhm.
right?
Yeah,
It's not just NIH funding that's going to
Mhm.
do that.
Yeah. that's
Should
true.
they be sharing in the profits with the local people, for example? Is there a way that they could share somehow from, that there could be a requirement that you're
Mhm.
going to give X% of your profits back?
Yeah,
or
I think
something?
that's a great thing to do. And Variant Bio, for example, is it Variant Bio?
Yeah, Variant Bio.
Yeah, they're trying to do that, and they
Mhm.
have this framework where they say we are giving so and so percent, I think 4% or thereabout,
Mm.
to share the profit, with the community that helped in the research. But then I always like to distinguish a little bit between benefit sharing and benefits of research
Mhm.
as a whole.
Mhm.
Benefit sharing, I try to put it more on that, when there is issue related to commercialization and all that, while benefit is for the conduct of the research alone, what do they stand to, community
Yeah.
engagement and all that.
Yeah, and one thing I'll just also mention is I had a discussion with the science editor. They're talking about even the possibility of requiring people when they write papers that are focused, let's say in Africa, that they have to say in their methods or somewhere what aspect of community engagement there is.
Yeah, okay.
So that'll be very interesting
Yeah,
if they
yeah.
do end up doing that. It's a way to sort of force people to have to
Mhm.
really think about that. I'm
Yeah.
so sorry. This is
Yeah.
like, oh yeah, go on, please.
Oh, well, I had a question that was related to that, which is, what about the major Western funding institutions? Do they have requirements as far as the tangible benefits, or is there thoughts that they might start requiring that for this kind of research?
Truly,
That's
there's no any framework for now. Or
I've
any
never been
requirement.
asked that ever
Yeah.
on
Yeah.
any funding. They've never said like, how is this actually benefiting people?
Yeah.
And have you done community
opposed to
engagement?
requiring it like on the journal, like the very end point is the
Yeah.
journal
Mhm.
requirements.
Yeah, they were saying that they might require it as part of the, if you're going to publish on
But-
those topics.
But by that point,
NIH, for example, doesn't request. Yeah. But that's a good point. I mean, those
Yeah,
are ways that you
absolutely.
could, like, if you tie it into funding, then you'll get people to act on that.
Exactly.
I think we have to end. It's a fascinating
Yeah.
discussion, but it's 10 after. I know there was a question from Mwangala, and what we'll do is she will get back to you by email. We'll keep track of that question. So thanks to everybody for joining us, and again,
Thank you.
thanks to our speakers. Right. Let's figure out how to undo this owl.
Stop sharing.
Can you unplug that right there?
This one?
Yeah. Or not the black one?
Yeah.
There we go.
Yeah.
I still don't even know if this thing's playing.
Hey, Bege,
I know.
we'll talk.
Anyway.
I actually have a question. With the,
Where's Dan? Where's Dan?
Oh, he's on the fourth floor.
Ah, okay.
Yeah, he's on the fourth floor.
there's some.
Okay. I just wanted to like
I used to
follow.
work in a lab where
Oh,
we
yeah.
look
But
at
he's on
functional
the third floor.
approaches to
Right
studying
when
business
you-
opportunities and
the
here's
elevator,
the
like
safety
doing,
function. So I was curious what kind of functional
first floor on the left.
approaches
Yeah,
are
you
you
see that
were you
there's like
going
office
to
space
use to
divided.
evaluate some of the variants that you discovered? It was mostly with the RAD 51C, right? And
Yeah, so because so those were slightly very specifically, so for those, initially we thought that we had, yeah, the RNA correlation. So we might